So welcome to our podcast. I'm Jazmin.
I'm Izzy.
And I'm Chloe.
So our topic for today was that we chose was the antidepressants, and we did choose a research paper that says the sex differences and effects of ketamine on behavior, spine density, and synaptic proteins in social isolated rats. This research was done in 2015 by Sarkar and Kajab. What they wanted to talk about was the relevance of depression among women to men, and they found out that it was twice as relevant among women compared to men, and women are more common to display these depressive symptoms. Different mechanisms that were used to explain this difference of depression being more common in women. Therefore, these researchers were interested in tackling these big questions. They explained the effects of isolated rats causing them stress and how antidepressants effects of ketamine work on depression. They examined the behavior of spine density and the molecular changes in the medial prefrontal cortex, also known as the MPCF, right?
FC.
There we go. MPFC between the males and female rats.
Alright, so how they went about doing this experiment was they started by putting the adult male and the female rats in either a paired house condition, which is just normal, regular living conditions, or in a solitary cage on a 12-hour light cycle each. So what they did to start was they tested the two groups for sucrose preference over a period of time. And the reason that they did this sucrose testing was because it's an indicator of anhedonia, which anhedonia is like a symptom of depression. So for this test, they habituated the rats to drink from two different bottles, and then before the testing even started, and then after a period of stress, then the rats were given the two pre-weighted bottles, so one with water and one with the 0.25% sucrose, and then they were able to choose. So the rats who preferred to drink sucrose had less symptoms of anhedonia, because if you have symptoms of anhedonia or just like uninterested in engaging in something pleasurable, it wouldn't matter to you which one you would drink. So once those sucrose preference levels were stable, so once the paired house conditions were choosing sucrose over water, and once the isolated ones were choosing just regular water, the groups were injected with either 2.5 or 5 milligrams per kilograms of saline, or 2.5 or 5 kilograms of ketamine. And then they tested them with the sucrose preference test again after 3 hours in giving them the injection, then 27 hours, and then on the third day they did the FST test. And the FST test, as we talked about in class, is the forced swim test. So that's a 2-day procedure where the rat is placed in a plexiglass cylinder filled with 25-degree water for 15 minutes on day 1, and then 5 minutes on day 2. And then they videotape their behavior, and blind scores then analyze their immobility time. And once again, immobility time is an indicator of how anxious they were, or feelings of depression in this case. So next, they also look at some biological things instead of just behavior. So they did a bilateral stereostatic surgery, and they infused them with HSV-GFP, which is a strain of herpes simplex virus type 1 that's tagged with a green fluorescent protein, so that they'd be able to count the spine density. And they did that in the medial prefrontal cortex, like Jazmin mentioned. And then 3 to 4 days later, they injected them with the different doses of either saline or one of the two different doses of the ketamine. And then they did immunohistochemistry, which is a technique for visualizing cellular components with GFP to detect the fluorescence, so that they could again count the spines. And then the spines were just qualitatively classified into 3 categories, being either mushroom, thin, or stubby, and they counted 12 neurons for each of the rat, or classified 12 of them. And then lastly, the rats in both the groups were sacrificed 1 hour after their forced swim test, and then they collected the brain tissues for a Western blot assay, and analyzed the synapsin-1 protein, the PSD95 protein, and glutamate-1 receptor.
Okay, so for the result, one interesting trend of the results was that for all of the tests where ketamine had any effect, the female rats responded to both doses, while the male rats only responded to the higher 5 milligram per kilogram dose. I'm just mentioning that right off the bat in case I don't mention the doses when I'm going through. That like holds out for all of them. So first, for the effects of ketamine on anhedonia and depression-like behavior in the isolated rats, the male rats started to show significant decline in sucrose preference in week 7, and they were considered stable by week 8. And then these effects were reversed by the higher dose of ketamine. The female rats, however, showed no decline in sucrose preference even after 11 weeks, and no changes in that were brought about by either dose. For the FST, in both male and female rats, isolation produced an increase in immobility time, which is the expected result. And a single injection of ketamine, both doses for females, only the higher dose for males, did reverse these effects.
Which, yeah, there's a lot of interesting things, like just in the one or a couple bit of results that we just discussed there.
Yeah, a big pointer that we should talk about is sample size. How do we feel about the way they had their sample done? I think how many female rats were there? 7?
Yeah, something like that. It's a very. I'm not really sure what would be a good sample size, but they said there were 6 per group, so it would have been 12 females in total. So 6 in the isolated, 6 in the paired house, and then there were 8 males
for each. So a total of 16.
Yeah, right. Okay. So I don't know, about 30 rats in total.
Yeah.
So I just don't know. I felt like I didn't know if that was a decent amount to have you know what I mean? Replicatable results. What do you guys think?
Do you have anything, Chloe?
Yeah. I agree. I agree with what you guys said. I don't recall seeing any red flags in their p-values or the little bits of statistical analysis that they put in, but then at the same time when you said we had 30 male rats, 30 female rats, that just sounds like it's kind of small.
But also on the other hand, that is you're caring for 30 rats, which obviously is a lot of work. So
yeah.
I don't know.
Even though it is a lot of work, I kind of felt that's where they lacked in having that sample size, because I'm thinking of comparing it with when we have human experiments and we take them in for research or something. We have hundreds and thousands of people that come in and out of the doors, and they take their samples or their research or whatever they're trying to find out, and for it to be 30 only, I feel is too little, and especially having more males, a total of 16, than females, which was a total of 12, which I said, I understand that, they do have to care for them, there's a lot of work invested, but I just felt it wasn't enough maybe to make a full experiment out of it.
Yeah.
Or a big conclusion at the end.
Yeah, it's definitely an interesting point to think about.
But like I said, I'm not a professional in this, right? So I don't know if that's a good size or not.
Yeah.
What about the timeline of how long they lasted each rat to, or they recorded each rat?
Yeah, I think it's so interesting that the female rats showed no decline in sucrose preference at all after being isolated for 11 weeks. It only took 7 for the males, and they even mentioned a little bit. Well, I guess we can't really be sure if by the time they hit the 11 weeks and they still didn't have did they still not have a decline in sucrose preference, and then they just called it that it was low enough, or it wasn't the same stability, I don't think, as the males. So right off the bat, we're already seeing this difference between the males and the females, just in their ability to resist, I guess, anhedonia symptoms. Yeah, exactly.
Yeah, it, like, when I first read that, it really made me wonder, because, like you said, we don't know for sure, but sort of like the impression that I got from reading it was like they got so far past the male rats that they kind of just had to like call it for the experiment. But like it really made me wonder, like, does that mean that like they wouldn't show any anhedonia symptoms, or is it just it would take much longer?
Like I even wonder if they went over the 11 weeks and like kept going until they saw, like what would have happened? Like how long does it take
To
the
really
female,
get the stabilize, the same stability,
than the male rat? So I don't know. I thought that's quite a difference.
Okay, so for the next chunk of results, these are the effects of ketamine on the spine density in the MPFC in male rats, and during the different phases of the estrus cycle in female rats that were subjected to isolation. The 8 weeks of isolation caused a significant decline in total spine density in male rats that was completely reversed by the higher dose of ketamine. When the morphology of the spines was analyzed, however, it showed that while both mushroom and thin types had declined during those 8 weeks of isolation, the ketamine only had a significant positive effect on the thin type and not the mushroom type. For female rats, it was a bit different because their estrus cycle had to be taken into account. So how the researchers did this was they tracked their estrus cycle and kind of like split them up into 2 subgroups. Those that would be injected during the de-estrus phase and those that would be injected during their pro-estrus phase. And so the isolation caused similar declines in total density as well as in particular the mushroom and the thin types in females as the males. One thing to note is that the total mushroom and thin densities were also lower in the rats that were in the de-estrus phase to those that were in the pro-estrus phase. And the ketamine had no effect on spine density for female rats regardless of their cycle or the dosage or their living conditions.
Yeah, so going along with that, the whole female rats and like, do we think that their estrus cycle was controlled well? I personally thought that it was because they make it seem, and I feel like they did, right, keep track of it. Like you said, they split it into subgroups and they were also injected. So I felt like there were so many variables into it that they had total control over that and how they can, even though there was that, I don't want to say error, but help me out. There was like that confounding factor, but like they still were able to keep that under control and still make it part of their research and be consistent with it.
Yeah, and I didn't include, like they did explain in the paper like exactly what like their methods were.
Right.
Yeah.
They were pretty rigorous.
I decided not to like include that in my explanation of the results, just because I was like trying to keep the results from like getting too out of hand.
Yeah.
But yeah, I definitely agree with Jazmin that their methods were pretty sound.
Yeah.
For keeping track of the estrus cycle.
Yeah, and going like off of that, what you were just saying, or how it was so much to consider when doing this experiment. So we talked about it in class a little bit, how a lot of times they don't even use female.
Yeah,
rats,
like it was recently
they just
told that they
doing
have to do it if you're gonna be making a research experiment.
Which I can see why, because it's such a huge factor, and like reproductive hormones, like that's like gonna affect your behavior and probably
It's
like
gonna
your
change a lot, to be honest,
neurophysiology and everything like that. So, and it's just something that you can't discount for either, because like you're never gonna have a female rat that doesn't have an adult
Their
female
cycle,
rat
yeah.
that doesn't have their cycle. And so you have to take that into account, and it really just kind of goes well with the validity of the experiment, which we'll talk about more later.
Yeah. And that's where we have to give credit, the fact that they did this and took the effort to take all these different variables and still keep control of them. So I thought that was really that was great that they included the female rats.
One thing that like I thought would be or like if I had more time or like more knowledge, I guess I would like to look more into the kind of what the significance are between like the mushroom and the thin type, and like what that really means, that there was more effect on the thin type and not the mushroom, like if those relate to certain behaviors or certain
like what does it really
neurons.
mean in a way. That's something that like I think is it would go into like another whole
experiment
to see what
that could be one within itself. All right, should we move on to the next set of results?
Okay, so the last set of results is the effect of ketamine on synaptic proteins in male and female rats subjected to isolation. So both male and female rats had reduced levels of the synapsin-1, PSD95, and GLU-R1 proteins. For males, the higher dose of ketamine reversed the decline of the synapsin-1 and PSD95 proteins, and it slowed down the decline of the GLU-R1 protein. But for females, again, as before with the spine density, neither dose really had an effect on any three of the proteins.
Okay. So for like the discussion, right, that was also part of our paper that we read. A big part of it was, I think, the experimental design, in which how it enabled like several differences between both the female and male rats. Even though they were exposed to the same stressors and the treatment paradigms as well, this really did help the results be more reliable, and especially when taking control of these variables that, like we brought up, like the cycles. They put the rats under the same stress, which was obviously something they took control of. The celerity cages under the IS of 12 hours, which you brought up, 12 hours of light/dark cycles. And the animal food was, I believe it was the same, and given the same amount to each rat, it wasn't like a difference of having an error, so that was good controlled as well. So they really did put a limit on their errors and what they had and what they detected. Another thing that they brought up was another research that was done with the female Wistar rats. Is that how it's pronounced?
So.
Yeah, okay.
So.
Which circulated levels of estrogen and can adjust the threshold of sweetness detection and the preference for sucrose. So that was like a confound variable between the two experiments that they compared theirs with. So
yeah, which might make sense then why that the females would have taken so much longer, because if those, like the hormone, the estrogen, that specific hormone, was kind of masking maybe their ability to be able to detect the sweetness, like they really couldn't even tell the difference between it. So like maybe it was more like a blind thing than rather not that they just didn't have anhedonia, maybe they just couldn't, like they weren't, the sucrose preference test wasn't an effective way to test their anhedonia because of their estrous cycle. So that is something to consider, and it makes it difficult because that was like the start of the experiment, and that's like the basis. So, like, there was like something
so we're questioning
that.
That was one of the like two, like they had like two behavioral tests, and that was one of them. And like based on this other research, it's like possible that
if we even question it, like kind of destroys that whole part. So. But it is an interesting point, though, that you have. Like, it could have been that for the reason that we couldn't detect the sucrose because of our estrogen level, or their estrogen level.
Right.
I think that's just like another reason why, like, it is important to, like, do these, like, types of experiments with female rats, because there are, like, so like with like the different hormone cycles, there are like so many things that can be affected. Like results wise, yes, but then like also just in like what like assays would be like best to use.
Exactly.
Yeah.
Definitely.
Okay, so like another point in the discussion that was a big takeaway from the experiment was how in the FST both sexes showed depression-like behavior, but males responded only to the higher dose, and whereas females responded to both doses. We saw similar results, or like similar trends, like throughout the experiment. Notably, male rats in normal paired housing conditions also showed lowering of immobility time on the higher dose, while females showed on both doses. And this agrees with previous findings that female rats are more sensitive to ketamine than male rats.
So what do you guys think about how females responded neurologically or like neurophysiologically, whatever you want to say, versus how they responded like to the behavior and like how ketamine affected that?
Yeah, I think that's probably one of like the most interesting things for me from the experiment was the fact that the female rats were more sensitive to ketamine, but at the same time, ketamine didn't
affect them.
Yeah, it's like they're more sensitive to it, but like it didn't really like always show an effect
Right.
on like certain things. It only affected like certain things, whereas like with the males were less sensitive, but at least it like consistently affected things.
Yeah.
Right.
Like Chloe said, because they were so sensitive and it did affect them in some ways, we would also, I would also make the conclusion that it would affect them throughout everything really, since they were so sensitive.
Right, so you would think if they were more sensitive.
Right.
Well, the thing is, like, they were more sensitive for behavioral things. So, for example, if they had decreased immobility time and stuff, or,
Yeah, the immobility.
and things like that. So
yeah.
they was able to improve or, like, reverse the behavioral effects of depression, but not the neurological effects of depression, which is interesting.
Yeah, they had it reversed the effects of immobility time, but then for the spine density, it didn't really show any effect for reversing that, and for the synaptic proteins, it didn't really show an effect reversing that.
It's just so opposite, and obviously, like, it could have just been, there are a lot of like experimental variables that we already discussed that like could have contributed to it, but it's so opposite to think that you could have, like, there was nothing going on in, like, there was no effect in the brain yet, like there somehow was behavioral improvements. You think it'd be the other way around, maybe there was some changes in the brain or something, but
especially since we talked a lot about in class how the brain kind of controls most of our behavior and all of that, so
or like maybe there's some sort of like different pathway that like ketamine affects that causes an effect for females that is different than in males.
Yeah, and it kind of goes into that, the next point that they did talk about. They mentioned that it really demonstrates that there's like some underlying difference, like you said, in the mechanisms for like the behavioral efficacy of ketamine between the males and the females, which is interesting to think that, I don't know, that there could really just be some underlying difference between them, which, obviously we know a lot of differences between male and female rats, but like, I don't know, you wouldn't really necessarily think that it would have that great of a impact on it. Yeah. So I feel like the main results from this experiment, you can let me know if you all agree, that this finding really highlights the sex differences in the effects of the chronic isolation on the behavior, the spine density, and the synaptic proteins.
Mhm.
The sex differences in how ketamine affects behavior, the spine densities, and the synaptic proteins, and then again with the estrous cycle, how those stage-specific differences in the female rats can really affect the spine density and different like mechanisms in the female rats.
Yeah, I think all 3 points are like the most
Blanket
Yeah.
kind of statements that you could
Mhm.
make.
out of like the whole experiment. I think those were the ones that were highlighted
Yeah,
pretty much. So,
I agree.
are there any limitations besides the ones we have already talked about, or further direction that this research could do? Like, I know that I took the further direction of like, I believe it could actually then, with all of the conclusions and if it was all correct and with the good sample size as we talked about, if that was all good, then I think you can then make the construct validity here of like being able to then put that research and compare it or make the conclusions also in females, which may be a big, step
Jump,
into it,
yeah.
jump to it. But I feel like if it was done well and if everything was correct, I think we could make those conclusions.
Mm-hmm.
Yeah.
Like it like we can compare that to the human behavior as well and how we would react.
Yeah, I agree. It definitely has like a big importance, and it's like a big hot topic, just because
Right.
there are very clear evidence that women are more commonly like display the depression symptoms
Mhm.
and things like that. And yeah, definitely that this might play a role into something we might not have thought about before, about how there really could be a huge underlying different mechanism between male and females, which goes into how maybe we need to figure out different treatment paradigms for
Yeah.
a male versus like a
Mhm.
female.
A female, exactly.
Which might get sticky.
Yeah, I don't, because this research was what, from
2015?
2015.
Yeah, so I don't know if anyone has like reproduced
Yeah.
these results or anything. We didn't really look into that. But yeah, I just think there were a lot of like really like interesting results from this study that like definitely deserve like further research.
Mm-hmm. Definitely. And we brought up like a few of them already before.
Yeah.
We talked about, the spine densities, even just like the estrous cycle, which they touched on. Like that could be
Yeah.
a whole experiment in itself. Yeah.
Overall, I would say it was a great experiment.
Yeah, I agree.
Like the only thing I would say, right, the critique would be the sample size. But other than that, like they took the female rats, they controlled all of their cycles, they did have reliable results and data, like they showed on their graphs, which were pretty complicated to understand, which was that's a limitation. Now I remember. Just because I am not that knowledgeable in this area just yet, I think that the one big limitation is that someone who doesn't know about this as much or didn't have like take a class like the one we're taking now won't really be able to know how to interpret the graph and like what it's talking about. At least that's how I felt. I feel like it was kind of hard for me.
I think like one reason that might be is because since they had two different housing conditions, and then they also had two different doses, and then they would also have to have like saline controls for both of those, and they put all of those onto like the
One
same graph. It's kind of hard to explain like verbally.
But yeah, they put all of their data with like just what Chloe said into one just figure. And so it's really dense. I would say you really have to break it up and follow through with it.
It's not something that you can like just like take a glance at and really understand the findings.
Yeah.
It's something that like you really have to like sit down and
it's just a dense visual display, but I don't know, it's not necessarily like a limitation, it's more just like a critique or a comment,
I guess. another great thing was that how they also compared like their studies with, they did their background research, so they didn't just like come up with this and they knew they didn't. And so like for the last like whole page or two, they added research of like who helped them out and how other researchers were brought up to help this come to life, I guess. Like they built on that.
Yeah, and like even when we were talking about like that one paper that was like, well,
The Wister rats.
The
or
Wister
whatever,
rats,
yeah, those like that, them bringing that up was definitely, it was really cool.
It's always helpful to use other information that's been done in the field to either support your studies or, you know, provide, again, like they did in that case, provide like a limitation that might explain why you didn't maybe get the results that you were expecting.
Yeah.
Which is really beneficial both to the reader and for anyone who wants to do further research, because then they know they have like places to jump off of. You know, they can be like, well, another person did this study, so maybe I can do something like this. You know,
And go into the mushrooms.
Exactly,
That you
yeah.
brought up.
yeah.
So yeah. Any other last things that we want to bring up?
Well, I think we may have already like kind of touched on this a little bit, but not really said it. But like one of the points is that like part of like the study design was that they had like a good incorporation of like behavioral tests as well as like physiological tests.
Yeah.
Which like really enabled us to, you know, see that like, oh, like the female rats, they are having behavioral differences, but they're not showing physiological differences for these like things that we're testing that are normally like physiological like markers of how the drug works.
Right. I definitely agree. That was like a really valuable, like you said, method for this experiment. Just because if you think about like what the results might have been interpreted as if they didn't do, if they just did the behavior or something, like just thinking about like the blanket statements that they would have made would have been very different than what they were right now. So it's definitely really important, and because you're never gonna have one without the, you know, your behavior without your physiological.
Yeah, they took the extra step to involve them both, which is great.
Yeah, I agree.
So, anything else?
I think that wraps it up.
Okay, so this is the end of our podcast. Once again, thanks for watching. Bye.